So chapter 14 is the brain and cranial nerves. We finished everything about the spinal cord, and now this is the brain. And the brain, if you still remember, the nervous system in general consists of central and peripheral nervous system. And the central nervous system consists of brain and spinal cord. We finished the spinal cord. So now the brain is cerebrum, cerebellum, diencephalon, and brainstem. These three together are called the brainstem. So cerebrum, cerebellum, diencephalon, brainstem. Don't forget that. The four main parts, and one of them is three. So cerebrum, cerebellum, diencephalon, brainstem. And the brainstem consists of mesencephalon, pons, and the medulla oblongata. The other name for mesencephalon is midbrain. Mesen means mid. Mesen means mid. Cephalon means brain. So mesencephalon means midbrain. So what are the main parts of the brain? Cerebrum, cerebellum, diencephalon, and brainstem. The brainstem is three parts: midbrain or mesencephalon, pons, and the medulla oblongata. So this was important. This part here is important as well. The development, you just need to know which is coming from which. So we start at the age of 3 weeks of the fetus with three parts only: pro, mesi, and rhombi. Means front and middle and back part. That's all. Prosencephalon, mesencephalon, rhombencephalon. So obviously the second part is the same of the name, but it is prosi versus mesen versus rhombi. So prosencephalon, mesencephalon, rhombencephalon, front brain, midbrain, and hindbrain, or the back brain. The prosencephalon will give you two parts: telencephalon and diencephalon. Mesencephalon will stay as is throughout the whole life. Doesn't change. Mesencephalon, mesencephalon, mesencephalon. That does not change. And it's also called the midbrain. Rhombencephalon will give you the metencephalon, myelencephalon. Metencephalon will give you the cerebellum and pons, and the myelencephalon will give you the medulla oblongata. This is how, and these are the parts that we were talking about in this slide. So this is what we have after birth, but the three give you 5 first. At the age of 3 and then 6 weeks, and this will stay as is at birth and throughout life. Now, if you still remember, the cranial meninges, or the covers of the brain, are from inside outside. What's the innermost? Pia. And what's the middle? And the outer one? Dura. Same thing in the brain. Same covers in the spinal cord are the same covers in the brain. It's the same thing. The outermost, which is the dura, is going to cover the brain. And anytime you have fissures or you have any depressions or something in between the different parts of the brain, the dura will make a fold. Okay? So it goes like this. For example, it goes like this. Here's the dura mater. And when you see a depression, it will dip in and reflect, making a fold. And these are called the dural folds. We have the following dural folds: falx cerebri or falx cerebri. Falx means the separation. So falx cerebri, what's the cerebrum? What are the main parts of the brain again? Cerebrum, cerebellum, diencephalon, and brainstem. Cerebrum, cerebellum, diencephalon, and brainstem. Cerebrum is the main part of the brain. This is the huge part. All other things are smaller parts, smaller units. So falx cerebri or falx cerebri, this is obviously the dural folds between the two cerebral hemispheres. So you have one cerebrum here, you have one cerebrum here, and there is a fold in between, separating the two parts. And this is called the falx cerebri. And within this dural fold, there are sinuses that are called superior and inferior sagittal sinus. And we will go through the sinuses later on, but just I want you to imagine if this is the dura that's covering the brain, okay? When it make a fold like this, in between the two parts, you see this space? The fluid will go within this, making a sinus. So within the fold, the fluid will go in, which is the cerebrospinal fluid, making a sinus. And this is the superior and inferior sagittal sinus, and you will see it later on coming. The next one is the tentorium cerebelli, and you have that in the lab. I don't know if you went through it or not, but the tentorium cerebelli means, tentorium means tent. Cerebelli means the cerebellum, the little brain, the small brain in the back. So tentorium cerebelli means the tent of the cerebellum. You know when you sit under the tent, the cerebellum is sitting under the tent. So this is called the tentorium cerebelli. So it's above the cerebellum and under the cerebrum. Tentorium cerebelli, and within this fold there is a transverse sinus. The next one is the falx cerebelli, and do not confuse cerebrum from cerebellum. Cerebellum with L, cerebellum is the little brain, cerebellum. Cerebrum is the big brain, the large one. So falx cerebelli, just like the same as we said when we talked about the falx cerebri, is separating the two cerebellar hemispheres. So we have two cerebral hemispheres and two cerebellar hemispheres. The two cerebral hemispheres are separated by falx cerebri. The two cerebellar, the little ones, are separated by falx cerebelli. So these are the dural folds. On the other hand, deep inside, between different parts of the brain and within the brain itself, there are some cavities. These cavities are for making, secreting, and recycling the CSF. And these are called the ventricles. So the ventricles are cavities within the brain that contain CSF, cerebrospinal fluid, the fluid of the central nervous system. So these are chambers or cavities, and these are lined by ependymal cells. And if you remember when we talked about the neuroglia, and it was in the exam, and I told you you will see many questions about this in the exam. And the ependymal cells are lining the ventricles because they make the CSF. What are the ventricles that we have? We have lateral ventricle. We have two of them within each cerebral hemisphere. See, here is one cerebral hemisphere. Here is the other cerebral hemisphere. The cavity or the chamber inside this is called the lateral ventricle, and another chamber inside this is called the lateral ventricle. So we have two lateral ventricles, and those two lateral ventricles are not communicating. They are actually separated by a partition that's called septum pellucidum. All what I'm talking about right now is in the lab. Either you did it or you're going to do it. The next is the third ventricle. We have only one third ventricle. So we have two lateral and one third ventricle. And this ventricle is the ventricle of the Diencephalon. This is the location. It's only one. And all these ventricles are communicating together. How? Lateral, the two laterals are communicating with the third ventricle through interventricular foramen or foramen of Monro. It goes like this. This is one cerebral hemisphere, and here is the other. This is one lateral ventricle inside. And here's another lateral ventricle inside. And this will be the third ventricle. And this is the fourth ventricle. So how many ventricles do we have?
3.
You can say 3, but the lateral is 2. So 2 laterals, 1/3 and 1/4. Why did we say lateral, third and fourth? They used to say first, second, third, fourth. But because those two are identical, so they stopped saying that. Now they say lateral, lateral, third, fourth. Instead of first, second, third, fourth. So 2 laterals, like the first and second, and then third, and then fourth. As you see, these ventricles are communicating, but the 2 laterals are actually separated. The 2 laterals are separated by septum. What's the name of that septum? Septum pellucidum. The transparent septum. There is a septum in between those. Now these ventricles are communicating with each other. They all contain CSF. And the CSF circulate within the brain to feed the brain, to give oxygen and nutrients to the brain. So it has to circulate all the time. Coming in, carrying oxygen and nutrients, and leaving, carrying carbon dioxide and waste products. And then circulate again, go to the blood and come back, carrying oxygen, nutrients. And then leave, carrying oxygen and waste products. The lateral ventricles are communicating with the third through interventricular foramen of Monro. Interventricular foramen or foramen of Monro. And this is the fourth, which is the next one. And the fourth is inside the medulla. So this, inside the medulla, this was inside the Diencephalon, and this was inside the brain itself, the cerebrum. Now the third communicates with the fourth through an aqueduct. What's aqueduct? Aqui means water or watery fluid. Duct, just a duct. So it's a duct that communicates aqueous or aqui, which is the CSF basically. So this is called the aqueduct of the midbrain, or the aqueduct of the mesencephalon. It's the same thing. So aqueduct anyway of midbrain. Okay? So again, the ventricles are there to contain the CSF, which is for nutrition and oxygen and removing carbon dioxide and waste products. How many do we have? We have 2 laterals, 1/3 and 1/4. Where is it located? Lateral within the cerebrum. Third within the diencephalon. Fourth within the medulla. These are all communicating because this is a circulation, just like the blood. So it goes from 1 to 1 to 1. From lateral to the third, this is called the interventricular foramen, or foramen of Monro. Between the third and the fourth, this is called the aqueduct of the midbrain. And this is a picture showing you all what we talked about right now. These are the 2 laterals. They communicate through the interventricular foramen, or foramen of Monro, to the third ventricle. And then from the third to the fourth, through the aqueduct of midbrain. And after the fourth ventricle, this is the central canal. What's under the medulla? Do you remember? Did you do this in the lab? Do you remember when you go with the midbrain, pons, medulla? What's after the medulla? Spinal cord. Yes, the spinal cord. So this is within the spinal cord. What is it? This is the central canal of the spinal cord. So the CSF is secreted here. Go to the third, secrete some more. Go to the fourth, secrete some more. Leave through the central canal, and it's actually going to circulate again and come back. We will see that later on. So from lateral to third to fourth to the central canal. Or from lateral to the interventricular foramen, going to the third, through the aqueduct, to the fourth, to the central canal of the spinal cord. So now you're circulating all the way from the top in the brain until the bottom in the spinal cord. Cranial meninges, PAD, as we talked about before. Here the dura mater itself is 2 layers. And the dura is the outermost layer, right? The dura is the outermost layer. So it goes like this. Pia, arachnoid, and dura. The dura itself is 2 layers. The dura is 2. The inner one is called the meningeal, and the outer one is called the endosteal. What's endosteal means? Endo-osteal. Endo means an osteum, bone. Yes. So this is at the inner aspect of the bone. This is what is is adherent to the skull from inside. That's where the name came from. So the dural folds, we talked about them, and they contain sinuses. We talked about this. And the ventricles contain CSF. Why do we have the CSF? Communicate between the different parts and carrying what? What does it carry? This is like the blood to the body. So it's carrying the good stuff to the brain and taking away the bad stuff away from the brain. What's the good stuff? Nutrients and oxygen. And what's the bad stuff? Waste products, carbon dioxide. This is what the CSF doing. It goes to the brain, giving them oxygen, nutrients, and take away waste products and carbon dioxide. This is one. Number 2, it also acts as a cushion. If you fall down on a water mattress, it's going to absorb the shock, right? So if you surround or put inside different parts of the brain something like a water mattress, which is the ventricles containing the CSF, containing fluid. If at any time you move, a vigorous movement or sudden movement or something, this is going to absorb the shock, to act as a cushion supporting the brain. We have choroid plexus, and the choroid plexus is what make the CSF and remove the waste products, and they produce about ½ liter per day. Okay, so what is the choroid plexus? These are plexuses, specialized ependymal and capillaries, ependymal cells and capillaries. They make CSF. They modify CSF. They remove the waste product from CSF. So it's all about CSF. Choroid plexus, CSF. So, and the choroid plexus, if you're following this, the choroid plexus will be within these ventricles. Like this. So it secretes within the ventricles, go to the next, secrete more, go to the next, secrete more, and so on. So choroid plexuses. From the choroid plexus to the ventricles to the central canal of the spinal cord. So this is the central canal of the spinal cord. From the central canal of the spinal cord, it's going to leak out or go out to the subarachnoid space. Do you remember the subarachnoid space?
Yeah.
What was the importance of subarachnoid space?
It's the space between the arachnoid and the tias.
Yes. What's the significance? What's the importance of that space? Take CSF, yes. So it goes from the central canal of the spinal cord to the subarachnoid space, which is under the arachnoid. And this is where we can get a sample of CSF from the subarachnoid space. From the subarachnoid space is going to circulate back. Now, choroid plexus secrete and modify CSF. How we get rid of the CSF? The CSF go to the brain carrying nutrients and oxygen. The different brain parts took the oxygen already. They take the nutrients. They started to produce carbon dioxide. They started to produce waste products. How are we going to get rid of it? It should enter and leave. It enter through the choroid plexus. How is it going to leave? It's going to leave through the arachnoid villi and arachnoid granulations. This is what's going to take it. Okay, are we following? Choroid plexuses within the ventricles secrete CSF. CSF secrete within the, circulate within the ventricles. Go to the central canal, subarachnoid space, and then it will go back. It is circulating inside. How to get rid of it? When it circulates, it's going to leave and go to the sinuses. The arachnoid granulation is going to absorb it and send it out to the veins. Go to the blood, to the heart. In the heart, it will carry more oxygen, nutrients, get rid of carbon dioxide and waste products. And then what? Then it circulates again. Go to the choroid plexuses. Choroid plexus take it from the good blood that contain oxygen and nutrients. Circulate within the ventricles. Go to the central canal, subarachnoid space, dural sinuses. Arachnoid granulation and villi will absorb it and move it out. Where? To the veins, to the heart. In the heart, you get rid of carbon dioxide, you load more nutrients. From the heart, it goes back to the choroid plexus, right? Are we following the whole circulation? One more time. Blood go to the central nervous system, carrying nutrients and oxygen. This is the good blood, okay? These, inside the ventricles, choroid plexus will take it, CSF, take it from the blood, carrying oxygen and nutrients, secrete it in the ventricles. It circulates within the ventricles. Feed the brain as we go. Go from ventricle to ventricle to ventricle, central canal of the spinal cord to the subarachnoid space. From the subarachnoid space to the dural sinuses. In the dural sinuses we have these. What are these? These are the arachnoid villi and arachnoid granulation. This is going to absorb it, take it, release it to the veins, go to the heart. Bring what to the heart? Bring deoxygenated and carrying waste products to the heart. The heart will deal with it. Get rid of the carbon dioxide, get rid of the waste products, get oxygen, get nutrients, and then go back, okay, to the choroid plexus. Choroid plexus take it from there, send it to the ventricles. Ventricles to ventricle to ventricle, to the central canal of the spinal cord, to the subarachnoid space, to the dural sinuses. Arachnoid granulation, absorb it from there. Take it out of the brain. Where? To the veins, to the heart. Heart, deal with it. Good blood, come back again, and so on. Did you get to the whole circulation? This is, and it keep on doing like that forever. Okay? Going back and carrying good stuff, leaving, carrying bad. Coming back, and so on. So these are the dural sinuses right here. And this is the subarachnoid space. So the granulation tissues are going to take the CSF from here, send it to the dural sinuses, and the dural sinuses will send it to the veins, to the heart. These are the different sinuses. Here is one, and here is two. And these are exactly like this. What section is this? Sagittal. So that's why they call it the sagittal sinuses. One of them is superior and the other one is inferior. So there is two like this. Superior and inferior sagittal sinus. Where's the name came from? Both of them are in the sagittal plane or sagittal section. One is superior and the other one is inferior, which is this. This is the superior sagittal sinus, and this one down here is the inferior sagittal sinus. Both of them are within the dural folds, falx cerebri, or falx cerebri. Okay, it will go from there to the straight sinus, to the transverse sinus, okay, and then it's going to go to the venous system after that.
What's the white part in between?
This?
Yeah.
This is the falx itself.
Okay.
So it goes like this. This is the white that you're seeing, and this is the sinus. Okay? So the white is just two layers adherent. When they separate from each other to allow the CSF to go through, this is called the sinus. So the sinus is within the fold. And depend on what the fold separating, this one is separating the two cerebral hemispheres. So you call it falx cerebri. The other one here, this, is separating the two cerebellar hemispheres. So you call it falx cerebelli. This one here is making a tent above the cerebellum, so you call it tentorium cerebelli, between the cerebellum and the cerebrum. Now the blood that goes to the brain, from which these choroid plexus will absorb the CSF from. Where is it coming from? It goes through the internal carotid and vertebral arteries. We have two sources. Each one of those is two. Internal carotid and vertebral arteries. How about the dural sinuses? Dural sinuses. It will leave through internal jugular vein. Internal jugular vein. So this is the internal jugular vein that bring back the blood to the heart. Internal jugular vein. If you look at this picture here, what is this? What is this? This part here. Which one? Do you remember the arterial system? Did you go through this? Like from the aorta, ascending aorta? Okay. It goes like this. This is the heart. Okay. The blood is going to leave through ascending aorta, aortic arch, descending. Okay, so this is the aorta, leaving the heart, ascending, transverse or arch, descending. This is a brachiocephalic trunk, giving you the common carotid and subclavian. Common carotid, subclavian. Common carotid give you internal and external carotid. Internal carotid enter the skull. You did the skull already, right? Do you remember the carotid canal in the base of the skull? This is where the internal carotid artery enter to feed the brain inside. On the other hand, if you look at this one, do you remember the transverse foramina in the vertebrae? Yeah, from transverse to transverse to transverse, this is where the vertebral artery go through. So this is why we have these foramina on each side of the vertebral column or the vertebrae. So for the vertebral artery to go up. So now what's happening here is, if you ever feel your pulsation here, okay, did you feel your pulse here before? If you put your finger here, you can feel the pulse. And if you see like in the movies or something, they usually do like this first, try to see if this person is still alive or not, and they do like this, they don't feel anything. Is he still alive or dead? How about this? If this disappeared, he's dead, because this is too close to the heart. If there is any pulsation here, he's still alive. The heart is here, and this is very close and very large. So as long as this person is having pulsation at this point, which is the common carotid artery right here on either side, and if you push a little bit, you can feel it. If you feel it, this person is still alive, even if he doesn't have pulsation here. So we usually go like this, feel the pulsation here. There is pulsation? Okay, that's fine. He's alive. No pulsation, don't say he's dead. No, how about this? If there is pulse here, even if it is faint, he's still alive. So this is the carotid arteries, right and left. Okay? It's going to divide. Okay? Outside is the external, feeding the face. Inside, or internal, it will enter and feed the brain inside. So you have two internal and two vertebral. So it goes like this. Are you imagining? Two internal carotids entering the skull, go to the brain. Two vertebrae from the back. So how many arteries feeding the brain? 4 altogether. 2 from here and 2 from the back. Those 4 are going to communicate with each other to make a circle inside the brain, and this circle is called the circle of Willis. Did you do this? Did you do the circle of Willis? Okay, you will do it. Here is the rationale. We hear about people having occluded, completely occluded carotid artery, one side. This is completely occluded. And they are still alive. How? Because the 4 arteries, you have 2 arteries in the front and another 2 arteries in the back. 2 internal carotid. Can you all see this? 2 arteries from the front, internal carotid, and 2 arteries from the back. These arteries are going to communicate with each other, making a circle, okay? Like a common pool. So, right internal carotid, left internal carotid, right vertebral, left vertebral. All of these together will pour the blood within a common pool. Why? Because this pool, which is called the circle of Willis, this is going to supply the brain with blood. So what if this artery is occluded? I think we heard about this, right? This is an old person who go to the doctor and he told him, oh, your artery is occluded 90%, right? Did you hear about this? 95%, it's almost gone. We hear about this, right? You have atherosclerosis. Did you ever check that before? Your artery is completely occluded, but he's still alive. And he can actually like walk, but he cannot run or do exercise. Why? If even one of these arteries is occluded, you still have other 3 sources that still pour the blood to this circle. And this circle will equally distribute the blood to the brain. So here is the thing. If each one of these supply part and one of them is occluded, this part will die, right? So that's not the case. So let's say 1 liter is coming from here, 1 liter from here, 1 liter from here, 1 liter from here. What's the blood? What amount of blood inside the circle? 1, 2, 3, 4. 4 liters, right? One of them is occluded. How many liters do you have? 3. But the 3 circulate within the circle. So you will give less blood. You get it? I have 4 of you. Okay? 4 of you. And every day I'll give each one of you a liter of water. Right? One day I received only 3. 1 liter is not there. Should I leave one of you thirsty, not drinking until he die? Or should I take the 3 and put them together and give each one of you 3/4 of a liter? Is that okay? Yes, you can do that. Why not, right? Give 3/4 of a liter better than giving 3 people 3 liters and let the 4th one die, right? So I'm going to take the 3 together, put them together, and I'll say, okay, let's share. This is what's happening. We share. Even if one of them is included, 3 are still working within this circle, and this circle is clinically very important. It's called circle of Willis. And it receives blood from 4 different sources, 2 internal carotid and 2 vertebral. Here is the circle I'm talking about. This circle here. So this is the internal carotid, another internal carotid, vertebral, vertebral. So all of them come together. You see this circle, complete circle here? Can you see this? It's a complete circle. So two vertebrals bring the blood to the circle. Two internal carotid. Here is one, and here is one. So four sources give blood to this circle, from which anterior, middle, and posterior cerebral are going to feed the brain. Anterior cerebral, middle cerebral, posterior cerebral. This is going to feed the brain. And all of them are coming from the same common pool, which is the circle of Willis. What's the CVA? Cerebrovascular accident or stroke? We hear this person had a stroke. What's a stroke? The stroke is after all of this that we're talking about, after the circle of Willis. What about a branch of these arteries coming from the pool got occluded for some reason? Or there's an accident or something. So the blood will shut off to part of the brain. This part of the brain is going to die, and this is a stroke. So what's a stroke or cerebrovascular accident? Cerebro means brain. Vascular means blood vessel. Accident. It's an accident in the blood supply to the brain. Cerebrovascular accident. An accident to the blood supply of the brain. One of the arteries got shut off for some reason. What are the reasons? Maybe a thrombus, blood clot. Maybe an accident that he lost the artery somehow. So this part that was receiving the blood is going to die, and this is what the stroke is all about. Now, when the blood goes to the brain, it doesn't go to the brain tissues as is. Not like the blood, not like every other tissues in the body. Every other part of the body except the brain will receive the blood directly. Like renal artery, give the blood to the kidney, right? Pulmonary artery, give the blood to the lung. Splenic artery, give the blood to the spleen. This is not the case in the brain. The blood goes to the brain, and then there is a barrier that separates the blood from the brain, and it will allow only certain parts to go through. Not everything go through. And this is called the blood-brain barrier. It's a barrier between the blood and the brain to protect the brain from harmful materials within the blood. So the blood goes to the brain, not all of it will reach the brain. Only part of it, fraction of it, will reach the brain. The part that's good, the part that the barrier allowed to go through. What can go through it? Lipid-soluble compounds. Oxygen can go through, enter. Carbon dioxide can go through, go out. Steroids, prostaglandins. What is controlling the blood-brain barrier? Astrocytes. If you still remember the neuroglia, I told you the neuroglia, very important. So astrocytes make or control the blood-brain barrier. Okay, and that's why you can take medication and they tell you, no, this is not going to affect the brain. Can I take antibiotics for the brain? No, it does not affect the brain. It goes everywhere else, but does not go to the brain. Why? It's not made to pass or to go through the blood-brain barrier. It has to be in a special composition to overcome the barrier. What's that composition? Lipid-soluble. They have to make it somehow to be lipid-soluble so it can go through. Blood CSF barrier, on the other hand, is made by ependymal cells. And this is in another spot, which is in the choroid plexus. This is where we make the CSF. Why do we have it? Just in case. If something go through and reach to the choroid plexus, even if it passed the blood-brain barrier and go to the choroid plexus, it will not be allowed to go through the ventricles. It will not be allowed to go to the CSF. And this is the blood CSF barrier. So we have two barriers. Now, the blood-brain barrier itself. It's not like a ball that's completely surrounded with a cover, because if that's the case, the brain will be completely isolated. And you cannot completely isolate the brain. Why? Because you have to allow certain things to go in and others to leave. So you have to have some breaks or some portion that is broken. You have to break it down in certain areas. Like what? And this is important. What are the breaks? Is it completely surrounding the brain? Is it completely isolating the brain? No. There are some openings in this barrier. What are the openings? One of them is at the hypothalamus, because hypothalamus is inside in the brain, and secretions from the hypothalamus need to leave. If you don't have a break there, how is it going to leave? So hypothalamus is one. Posterior lobe of the pituitary gland is another one. Because of these secretions, ADH and oxytocin, they have to leave the brain and go out. So this is another one. There is a third one for the pineal gland, and there is a fourth one for the choroid plexus. So what are the 4 breaks of the blood-brain barrier? Hypothalamus, posterior lobe of the pituitary gland, pineal gland, and choroid plexus. 3 glands and 1 plexus. Hypothalamus, posterior pituitary, pineal, and choroid plexus. 4 breaks. 3 of them are for glands and 1 for choroid plexus. Now from this part, we need to know the basic function of each part of the brain. What are the parts again? The main parts of the brain. Cerebrum, cerebellum, Diencephalon, and brainstem. What are the 3 parts of the brainstem itself?
The pons, the medulla,
And
the midbrain.
midbrain. And it goes like this: midbrain, pons, medulla. You need to know the arrangement. Midbrain up, the upper part. Under the midbrain you have the pons. Under the pons they have the medulla. What's other than the medulla again? Spinal cord. Okay? Midbrain, pons, medulla, and then the spinal cord. So what are the 3 parts from above going down? The 3 parts of the brainstem?
Cerebrum.
Parts of the brainstem, not the whole brain.
Oh, midbrain.
And what's under the medulla? It's not part of it, but what's under? Spinal cord. Okay? So now, the function will be as follows. One function, just to make it easier, one function is about the location. So if I ask you, like, what's the function of the pons? Midbrain, pons, medulla. To communicate the midbrain to the medulla. Yes. What's the function of the medulla? To communicate the pons with the spinal cord. Yes. What's the function of the midbrain? To communicate the pons with the cerebrum. This is the first function, which depends on the location. Okay? And this is like common sense. It goes without saying. Okay? Like if I'm here and I'm doing many functions, what's the first function? I'm communicating those two, the 2 students here. I take something from you, give it to her, or the other way around, because of my location. Plus special functions that we need to remember. What are the special functions? And I will focus more on the most important part that you have to remember. When we talk about the medulla, number 1, you have the autonomic nuclei that control the visceral activities. What are the visceral organs that are controlled? What do you need to control within your viscera? Anything in here. All your viscera are in the trunk. Anything. Okay? You're talking about the lung, you're talking about the heart, you're talking about the viscera, you're talking about the intestine, anything in here. So the nuclei. And what's the difference between nucleus and ganglia?
Nucleus is inside the spinal cord.
Inside the not necessarily spinal cord. Inside?
The central nervous system.
Yes, inside the central nervous system. And the ganglia is outside. And what is it? Collection of?
Cells.
Bodies, specifically. The body of the cells. Collection of bodies. So these nuclei, which is equivalent to the ganglia outside, these nuclei are, so in each part of those, this is what I want you to know. You have to know this. In each part. The first function, which is the simple one, is all about the location. You are in between different parts. You're communicating these parts. This goes without saying. The special function is what you have to remember. It's always about nuclei. Nuclei do the function. So what are the nuclei of the medulla? You have autonomic nuclei. Autonomic nuclei contain the visceral organs. Number 2 will be roots of cranial nerves. They will make the cranial nerves. So in each part of this, we're talking specifically about this. What is this? This part only. Brain stem. What is this? And what is this? And what is this? How many cranial nerves do we have?
3.
Cranial nerves. The nerves that, how many spinal nerves? We finished that already.
2.
Spinal nerves, all the spinal nerves, all these.
Oh.
31. 31. How many cranial? You have 12. Okay? Those 12 will come out of each part of these. So the function will consist of 3 parts. For each one of these, you have to remember 3 parts. Number 1, location. Because of your location, so you're able to communicate different parts to each other. That's 1. Number 2, which cranial nerve come out of this? We will cover it. Number 3, the nuclei. Okay, so for each one of those, the function is 3 things that you have to know. Location. Number 2, nuclei. Number 3, cranial nerves. Which cranial nerve arise from this? So cranial nerves will go like this. Okay, which cranial nerve come from which part? This is what we have to know. So nuclei of the medulla contain autonomic nuclei, visceral activities. Most importantly, that you have in the medulla, beside the visceral, controlling the visceral activities, is the nuclei controlling the lung. Okay? You breathe, how many times do you think you breathe per minute?
5, 6.
20, 14, 16. Yes, it's not a fixed number. Okay? It's somewhere between 14 and 20. Okay? 12 and 20. Anything is okay. Each one of us is different. Maybe I'm 15 and you're 18 and he's 13 and she's 15. Yes, anything in that range is fine. What decided that rhythm? It's the nuclei in the medulla or the medulla. Okay? They set the base, the basic rhythm for the breathing. So it's called the breathing nuclei or the breathing centers. Okay? So what are these nuclei that we have to know? Cardiovascular and respiratory rhythmicity centers. This is what I was talking about. Respiratory rhythmicity. What's the rhythmicity? The rhythm. What's the rhythm? How many times per minute? Okay? So this is in the medulla and the cardiovascular centers. Cardiovascular, that control the heart and the blood vessels. Okay? This is one. There is also reticular formation inside the medulla. And what I want you to remember about the reticular formation is just regulation of the autonomic functions. But the most important thing to remember, the most important, is nuclei controlling the cardiovascular or cardiovascular centers, rhythmicity centers. This is one. And the cranial nerves that come out of this, and I will talk about it. 9, 10, 11, 12. 9, 10, 11, 12. When you see all the cranial nerves, you can remember them all together. Okay? So the last, 8. Sorry. 8, 9, 10, 11, 12. So the last 5. You have from 1 to 12. 8, 9, 10, 11, 12. The last 5 come from the medulla. we have other nuclei, but I'm covering the most important ones. The next part is the pons. What do you need to know about the pons? The pons will have other centers, cardiac centers, respiratory rhythmicity centers, and vasomotor centers. So cardiovascular and respiratory rhythmicity. But isn't that exactly what we said about the medulla? Respiratory rhythmicity centers in the medulla. There are other respiratory centers in the pons as well. So there are respiratory rhythmicity centers in the medulla and others in the pons. What's the difference? Here is the difference. Those in the medulla set the basic rhythm. Okay, so this is called the respiratory rhythmicity center. This is what decides you're going to breathe 15 times per minute, 16 times per minute. How about those in the pons? You have respiratory centers also in the pons? Yes, but the pons, because it's above the medulla, it's going to modify. So the medulla will say 18, okay, but the pons will say, okay, I understand that you want it to be 18, but now we're doing more effort. Make it 25. Okay, so those respiratory centers in the pons had an upper hand on those on the pons, on the medulla. Now the cerebellum. What do you need to know about the cerebellum? Cerebellum, which is here, you need to know position, balance, movement, and muscles. Okay? If you remember those, that's all what you need to know. It controls your position, your balance, your movement, and your muscle tone. Okay? BPMM. This is a function of the cerebellum, and this is what we have to remember. Okay? It controls our position. It controls our balance. It controls our movement. It controls our muscle tone. BPMM. This is what we have to know about the cerebellum. And I'm summarizing, giving you like the basics, and this is good enough to remember. Now the diencephalon. What's the diencephalon? The diencephalon is basically the thalamus and things around the thalamus. So it's basically thalamus, epithalamus, and hypothalamus. Thalamus, epithalamus, hypothalamus. Thalamus, what's above the thalamus and what's under the thalamus. Epithalamus, thalamus, epithalamus, hypothalamus. The epithalamus contains pineal gland that secrete melatonin. Okay? So we're talking about the function of each one of those. What's the diencephalon? Again, what's the diencephalon? The diencephalon is thalamus. Above the thalamus, under the thalamus. Thalamus, epithalamus, hypothalamus. Thalamus, epithalamus, hypothalamus. What's the function of the epithalamus? Contain the pineal gland that secrete melatonin. How about the hypothalamus? The hypothalamus is kind of complicated a little bit. The hypothalamus will contain different bodies, and each body has a function. Mammillary body, control feeding. Autonomic centers control the nuclei in the medulla. Supraoptic nuclei secrete ADH. Paraventricular nuclei secrete oxytocin. Preoptic area regulate the body temperature, and suprachiasmatic will control the day-night cycle. So there is some more details that we need to know about the hypothalamus. So the diencephalon is thalamus, epithalamus, hypothalamus. Epithalamus contain the pineal glands that control, that secrete melatonin. How about the hypothalamus? The hypothalamus contains these nuclei. And each one of those has a function. Mammillary body, feeding. Autonomic center, control the medulla. Supraoptic, ADH. Paraventricular, oxytocin. Preoptic, body temperature, regulation of body temperature. Suprachiasmatic, day and night cycle.
Do we need to know all the functions?
Yes. What I'm saying right now. One word. Mammillary body, feeding. Autonomic center, medullary nuclei. Supraoptic, ADH. That's what you need to remember. Supraoptic, ADH. Paraventricular, oxytocin. Preoptic, body temperature. Suprachiasmatic, day and night cycle.
How about the two groups?
This is what secrete the hormones. Don't worry much about it. We have a system inside the brain that's called the limbic system. And you need to remember two words about the limbic system. This is what I want you to know. Limbic system. Limbic system. Remember the M of the limbic? That will give you two things. Memory and emotion. Can you remember this? Limbic, M, memory, emotion. That's all. Just remember this. What's the function of the limbic system? Limbic, M, memory, emotion. Can you remember this? Memory, emotion. Limbic, memory, emotion. Now the white matter in the cerebrum. We're talking about the cerebrum now. The cerebrum itself, which is the main part, the big part. When we talked about the spinal cord, we said that the gray was inside or outside?
Inside?
Inside, and the white was outside. Here it's the opposite. The inside is white and the outside is gray. Okay? Inside is white and the outside is gray. The outside is the cortex. I think you started the central nervous system already? The neuroanatomy in the lab?
Yeah.
Didn't start yet? You did? The cortex is the cover outside, and this is a gray matter. The filling inside is white. What is the white? The white contains different fibers called association, commissural, and projection fibers. Association fibers are the fibers that connect within one hemisphere. This is the right hemisphere, this is the left hemisphere. Within one hemisphere, the connection between different parts is called association fibers. Short fibers, like from gray to gray, this is called the arcuate. And the longitudinal ones that connect one lobe to the other. I don't know if you got through that or not, but if this is the brain, if the brain is like this, okay? The brain consists of different lobes. Frontal, parietal, occipital, temporal. Did you do that? Okay, if you're connecting lobe to lobe, this is called the longitudinal fibers. Okay? If you're connecting gyrus to another gyrus, this is called the arcuate fibers. Okay? So, and all of this is within one hemisphere. I didn't talk about from one to the other hemisphere. We are still within the same hemisphere. Okay? So within the same hemisphere, when you connect gyrus to gyrus, this is called the arcuate. When you connect lobe to lobe, this is called the longitudinal fasciculus. The next part is the commissural fibers. And this is what actually communicate the two cerebral hemispheres with each other. And this consists of corpus callosum and anterior commissure. You will see all of that in the lab if you didn't see it yet. So these are called the commissural fibers. So what are the association fibers within the same hemisphere? Commissural fibers between the two hemispheres, connecting the two together. Corpus callosum and anterior commissure. The third one is projection fibers. This is actually connecting the brain to the outside. So we have three types of fibers again. This is your right hemisphere, this is your left hemisphere. Within one, the same hemisphere, the fibers that connect different parts in the same hemisphere, this is called association fibers. The fibers that connect the right and left hemisphere, commissural fibers. The fibers that connect the hemispheres, all of them, the whole cerebrum, with the outside, this is called the projection fibers. And the projection fibers will link the cortex with all different parts of the brain. So it goes like this. Connect from the cortex to different parts, like this. So it actually goes outside of the cerebrum. So if you look at these, what is this, the blue? Connecting gyrus to gyrus, arcuate. This is connecting one lobe to the other lobe. This is the longitudinal fasciculus. This is between the two hemispheres. So what is it? Commissural, right? Yeah. Like the corpus callosum and anterior commissure. Okay, if you imagine the brain like the watermelon. Imagine the brain like a watermelon, seeded watermelon. You see that the watermelon is outside is green, right? The filling inside is red, and you can have some seeds, black seeds, right? This is how the cucumber looks like, the watermelon looks like. Oh, the cucumber, it's the same thing. So the outside, the green, is called the cortex. The inside, which is the red, this is the white matter. The black seeds inside, these are called the basal nuclei. Okay? So what are the basal nuclei? These are nuclei inside the white matter. What's the function? Subconscious control of the skeletal muscles and coordination of the learning movements. So when you learn different movements, like this is a baby who is 1 or 9 months, for example, and he started to learn how to walk a little bit and like stand up and fall down and stand up. What he's doing through this period, he's training his basal nuclei. Okay? Until these basal nuclei are trained, then he can walk and other movements. Not this is for the babies, but for everybody else, if you wanted to learn a movement, like you're learning how to play guitar, for example, or how to play piano, you at the beginning it's really rigid and you cannot do the movement that easy. But when you train your basal nuclei, this will acquire this ability to coordinate the movements. Now the cortex itself, the cover, the green outside. Each one of those will have a function that is very important to know. Special functions, special sensory cortex. What's the special senses? What are your special senses? Hearing, smelling, taste. What else? Sight, vision. Yes, these are the special senses. For each one of these senses, there is a part in the cortex that's responsible for this function. For the visual cortex, and this is located here in the back, on the occipital lobe, this is the part of the cortex that's responsible for vision. The next part in the temporal region, this. And to remember it, where is your ears? What is the lobe that's right beside your ears? Which lobe is here? Temporal. So the temporal lobe contains the auditory cortex. Then you have the olfactory and gustatory. What's gustation or gustatory? What does it mean? What's gustation or gustatory?
Taste.
Taste. Okay? So this is what contains the taste. So here's the occipital lobe that contains the visual, right here. The olfactory cortex is inside here at the insulate, and so as the gustatory. So both olfaction and gustatory, smelling and tasting, both of them are located here. The visual is in the back here, and the auditory is in the temporal lobe. Okay, so now let's say when you touch something, okay, you don't know what it is at the beginning. You touch something you don't know, right? But if you touch the laptop, for example, okay, and you see it, and you have a memory of it, next time when you feel it, if you close your eye and feel it, you touch it like this, can't you tell that this is a laptop? When you touch like the keyboard and the screen, can you tell? This is called the association area. So the first area that receives the sensation is just the basic function, okay? You see something and you don't recognize what it is. Like a baby, for example, is seeing something, okay? See the laptop, for example. What is this? He sees something black. He don't know what it is. But you tell him, this is the laptop. This is a laptop. Okay? Later on, he will acquire this in his memory, in his association area, and next time when you see it, he will associate this look with what you told him. So when you see this, this is a laptop. When you see this is a car. So next time, he don't say, this is a something that's moving. No, this is a car. How did he know? Association centers. So each one of these cortex that will receive the basic function will have an association area surrounding it. Another example, you hear something in the beginning and it sounds like something. You don't know what it is. Music. Yeah, what type of music? Just music. I don't know. Just music. Okay? So I tell you, okay, listen to it again. And this music is jazz music, for example. Okay? So when I tell you that, you will make a memory that this type of music is jazz music. Okay? Next time when you hear it, what is this? You don't say it's music anymore. You say this is jazz music, right? How did you know? It's not about the basic one. The basic one, receive it as something, roughly speaking. This is music. This is a sound, something. Okay? It's a noise. But the association area that's accompany the auditory cortex will tell you exactly what it is, but not from the first time. The first time you see it, you don't know what it is. When I tell you, maybe once, twice, or something, the association area will keep a memory for what is this. And next time, you can tell exactly what it is. And this is the association areas. And we will stop here and finish the rest next time.